like we have completed before starting the class, I want to let you know that we have completed with the genetics part first, means heredity and variation part, Mendelian part, right? So you can refer with the videos and all that. You can contact with the team. And the thing which is going on, we have started with the molecular genetics. The reason behind it, starting it, that it have major role in the medical examination, right? In the NEET examination, approximately you will get the 18 to 20 question from this genetics part, including both of the chapter. So if we, let's say, if we talk about the marks percentage, so you will get the approximately 70 to 80 marks question from the genetics itself, right? So it's very important. So the only thing which is left with that, there are three topics which are left in this genetics, complete genetics part, like in second chapter, molecular genetics. So today we are going to start with the operon. Fine. So what is the operon, right? So what is the operon? Basically, operon, lac operon is a model which represent how does the gene function, right? How does the gene work, right? The functionality part of the gene. The functionality part of the gene means the mechanism by which the gene function. It was represented by two scientists, Jacob and Monod. Jacob and Monod, they worked on the bacteria, and actually they work on the gene which is responsible for digestion of lactose sugar, right? That's why we call this model lac operon model, right? It is also given in your NCERT book. So let's discuss how the lac operon works. Before starting this operon, I want to tell you one thing. The bacteria, lactobacillus bacteria, have this operon, lac operon, and there are some other bacteria also which have this gene set, right? When there is a lactose in medium, suppose that a bacteria is there and bacteria have lactose sugar around it, then the bacteria will release the enzyme and digest the lactose sugar, right? The part of the gene, which is responsible for the digestion of lactose sugar is known as the structural gene part of the lac operon. So look, actually, this is the structural gene, LAC-Z gene, LAC-Y gene, LAC-A gene. LAC-Z gene, why I am telling you, and why I have written here, because this question have already been asked in medical examination, that LAC-Z gene is responsible for the release of, now there are four options. You have to remember this thing, like which gene is responsible for a production of which enzyme, right? So look, this is a structural gene, means this is the part of the gene which code a particular enzyme. That enzyme digests the lactose sugar, right? LAC-Z gene is responsible for release or secretion of beta galactosidase. LAC-Y gene is responsible for release of permeases enzyme. LAC-A gene is responsible for trans-acetylases, right? Release of trans-acetylases. LAC-Z, LAC-Y, and LAC-A gene is called structural gene. The part structural gene, what is a structural gene? The part of the gene which code for particular amino acid or polypeptide, and that polypeptide or amino acid transform into enzyme, right? So these genes are called structural gene. So when there is lactose in medium, then this gene will start functioning, means the gene will switch on and it will produce enzyme. Suppose that the lactose is not present in medium, then the enzymes are not required. When the lactose will not be present in the medium, on that time this gene will get switch off. Now how this gene will switch on and how they switch off, right? This mechanism is called lac operon. So look, the lac operon include these all genes which is visible in your screen. First, I will start from the D diagram. When there is no lactose, right? When lactose sugar is not there. When lactose sugar will not be there, so look, this is the structural gene. This part of the gene, this red color part, right? This red color part is called operator gene. Operator gene will switch on the functionality of a structural gene or switch off whenever it is required. So operator gene is a type of switch. If this switch is on, then the structural gene will function and formation of enzyme will take place. If this operator gene is switch off, then this structural gene will not function and formation of enzyme will not take place, right? Next, there is a promoter gene. After the operator, there is a promoter gene. This promoter gene is that part where the RNA polymerase enzyme will come and join and start transcription. At this some distance, vicinity, right? Not too much distance. At this some distance, there is a one more gene, regulatory gene. Regulatory gene is the gene which is responsible for switch on or switch off of operator gene, right? So this complete set of the gene, this is called lac operon. Now come to the first B model. Why I am talking about the B model? Because this is at this condition, the gene is switch off. When lactose is not present, then this gene will switch off. What will happen when lactose is not present in the medium around the bacteria? This gene, what this gene will do, it will code for a protein, right? So this is a gene which is responsible for coding of a protein. So this regulatory gene will produce code for a protein. This protein, the name of the protein is a repressor protein. This repressor protein will suppress the rest of the gene. How it will suppress? Look, this repressor gene will form a repressor protein. This repressor protein will go and fit over here, where it will fit over regulatory gene. When it will fit over here in the operator gene, when it will fit at the operator gene, it will block the this part, and now the genes will be get switch off. Now what will happen in this gene, in the promoter gene, RNA polymerase will come, it will join, right? It will join, but it cannot go ahead. Why it cannot go ahead? Because there is a hindrance, there is an obstacle. What is the obstacle? Repressor gene. This repressor gene is obstacle. It will not allow RNA polymerase enzyme to go ahead, and due to which there is a no transcription. If no transcription, no protein formation will take place, means no polypeptide formation will take place, and no enzyme formation will take place. Is that clear or not? Please tell me.
It's clear, sir.
Yeah. Okay, fine. Now, suppose that if there is a lactose, you have a milk and you have put this bacteria in that milk, right? Now, lactose sugar is present, because lactose sugar is sugar which provides a sweetness to the milk. Now, what will happen? Now, lactose sugar is present. Now, the enzyme secretion will take place. Now, what will happen? Again, the regulatory gene will produce, it will transcribe, it will produce a lac repressor, right? It will produce a repressor protein, right? This is active site. Now, this lactose sugar will come and it will join at the active site of the repressor. It will change the structure or conformation of this repressor protein. It will make it, after joining it, after binding with it, it will make it inactive. When it will make, which will make inactive, this lactose itself will make it inactive, right? Lactose will join at active site. While joining at active site, it will make it inactive. When it will make it inactive, at this condition, it will not join to the operator gene. When it will not join to the operator gene, now the gene is switch on. What will happen? In the promoter gene, RNA polymerase will come, RNA polymerase will join and start the transcription, means start moving from 5 to 3 prime end. When transcription will take place, we know formation of mRNA will take place. When mRNA will be formed, then what will happen? Formation translation will take place and formation of protein will take place. This is the functionality. That's how the lac operon works, right? Is that clear or not?
Yes, sir.
Abhinav, what's about you? Are you getting?
Yes, sir.
Okay. And Alia, are you getting? No? Okay. Okay, you are messaging me. Okay, very good. Good, good. Right. So what you have to do, actually, look, what I'm telling you right now, what exactly you have to do during the crash course, you have to take this screenshot, and whatever I say that this is the question, that is the question, what can be the question from this one? Now the question is that, what can be the question? Which inactivate the repressor? So lactose inactivate the repressor gene, right? What is the function of operator gene? What is the function of regulator gene? What is the function of promoter gene? What is the function of lac Z, lac Y, lac A, right? These can be the question. That's what you have to remember it, right? This is the only question in this, right? Please take the screenshot, then I will go ahead. Next concept. And yes, in the previous year question, you will see that my team have already forwarded you that PDF of previous year question. You will find that in previous year older questions, even it has been asked who have discovered the lac operon. But currently, you won't get these kind of the simple question that who have discovered the once in a blue moon you can get, but normally you won't get that much simple question. The question can be what lac Z produces, what lac Y code for, lac A code for, what is the structural gene which is not the part of the structural gene, right? That kind of a question. So this image should be in your mind, right? Clear? Please take the screenshot. Also this one. So this was the lac operon. Now one more thing, Human Genome Project. I have only incorporated that part which have been asked as a question. So what is the Human Genome Project actually? I will brief for you. Actually, in Human Genome Project, humans have number of the genes, right? All those genes are present in the 23 pair of chromosome, right? So first scientists have arranged first pair, second pair, third pair, fourth pair, and all that. By this way, from first pair to 23 pair have been arranged in this way, right? Then what scientists did after doing this, what they did, they arranged that in the first pair of chromosome at the first position, this gene is present. At the second position, this gene is present. Third position, this gene is present. So that's how they have arranged. So as we have discussed at the number of the time, that the place where genes are found, the place is called locus, right? And loci is plural word, right? So at first position, if suppose that hair color gene is present, then we'll say at the first locus, in the first chromosome, first locus, hair color gene is present. As we can say in the 23 pair, 23rd pair, right? At the second position, gene is present for the health of the male or female, right? Because 23rd pair is sex chromosome, right? So that's how what scientists have done. Scientists have in Human Genome Project after the number of years and at this when the billion of the rupees is spent, then the scientists came in the conclusion that which gene is present at what location and what is the function of that. So this complete project is called Human Genome Project, right? Now, what was the question? What can be asked as a question? The total number of the gene is 3,164.7 million. So there are 164.7 million genes are present in a human being. The average gene means a single gene. An average gene of a human being have 33,000 bases, right? The largest gene, this question have already been asked multiple times. The largest gene is responsible for formation of dystrophin. Can anybody tell me what is the dystrophin protein? Where it is from? Dystrophin.
So can I repeat the question?
Question is, Iram, that dystrophin protein, what is the function of dystrophin? What is the function of dystrophin? Basically, this dystrophin is responsible for muscular functionality of our muscles, right? So, largest gene is dystrophin, and it have 2.4 million bases, right? Next, the total number of gene in human genome are 30,000. The maximum, this question, even this question I've been asked in this one. The maximum number of gene, 2,964 gene are found in chromosome number 1. Next question, next concept also have been. The minimum number of gene are found in the Y chromosome. They are only 231, right? Got it? Take the screenshot of this one, human genome project. So that was the human genome project. It was a factual part, so I think everything must be clear with you. Now, DNA fingerprinting. Can anyone tell me what is the DNA fingerprinting? What is the purpose of DNA fingerprinting? Tell me, Abhinav. What is the DNA fingerprinting?
Sir, it is the process of like you remove the strands of the DNA and verify the proteins.
Remove the strand of DNA and verify the protein. Alia, what about you? Hiram?
Sir, you're asking the users or like what it is?
What is the purpose of DNA fingerprinting?
We can like use it to identify like dead bodies,
Yeah, we can.
and then it's used in court cases for like
Exactly.
understanding your parents and all.
Right. This is the purpose, and
And,
basically, yeah, you're right.
in criminal cases.
Exactly. It is used in crime scene salvage. Look, the function of the main purpose of DNA fingerprinting. We can define that who's the actual biological father or biological mother of a particular offspring, right? If there is a particular child who's actually the actual means biological parents, right? Then that can be, and it is used when at the cases of the property cases, at the cases when there is a son or a daughter, number of the mothers they claim that this is my daughter or my son, right? On that time, and sometime the son or daughter they want to say that this is the particular person is my father or my mother, and on that time the court says for go for DNA fingerprint. And what is DNA fingerprinting? And one more thing, suppose that there is a crime scene, somebody have been murdered, right? So, and before the murder there was a fight between the person who killed uh, who killed that person, right? So on the fight, what happened, the hair falls down, or the rest of the skin or the part of tissues they remains in the nails during the fight, right? So scientists take a forensic scientists take out that part of the DNA, and then they matches the DNA of the suspect, right? So that is also used in forensic science. In the nutshell, we can say DNA fingerprinting is responsible for to prove the biological relation of a particular person, right? So suppose that if you will take my DNA fingerprinting, the 50% part of that DNA sequence will match with my father, 50% part will match with my mother, right? And even it will 25% it will match with my grandfather or grandmother as well, right? So that is the DNA fingerprinting part, right? Even the 50% part of the DNA sequence that will match with my son as well, right? So this is the DNA fingerprinting. So what exactly we do in DNA fingerprinting? Please try to understand whatever I am trying to say right now. In DNA fingerprinting, look, this is a structure of a chromosome. This is an arm of chromosome. This one is an arm of chromosome. This is another arm of chromosome, right? And this is called primary constriction. This is called secondary constriction, right? After secondary constriction, there is a satellite part, right? This is satellite part. In satellite part, so this what is the difference in the satellite part? The specialty of the satellite part that it have repetitive sequence of the DNA, multiple repetitive sequence. So suppose that there is a gene which is 600 time repeated, right? So these repetitive sequence are called VNTR, right? So suppose that there is a set of gene that is, let's say it is a ATGCCGA. This is a gene. Now this gene is repeated 600 times. So if this gene is repeated 600 times in me, then it will be repeated 300 times in my mother as well, right? So 50% this repeated sequence will match with my mother, and rest of the 50% repeated sequence will match with my father, right? So these repetitive sequence are called VNTR, variable number of tandem repeat, right? So in DNA fingerprinting, we matches the VNTR, variable number of tandem repeat. This 50% VNTR should resemble with father and 50% VNTR should resemble with mother. This process is called DNA fingerprint. Tell me, is it clear or not? VNTR is the question of. And satellite DNA. Satellite DNA have been asked multiple times. Satellite DNA and VNTR, two are the question from this heading. Is that clear? Iram, Abhinav, Alia?
Yes, sir.
Yes, sir.
Very good. So, great. So this was the, I think this was the last topic. Yes, DNA fingerprint printing was the last topic. Okay. Now, previous year questions. Let me show you some glimpse of previous year question. As usual, what we do. So look, it is already tick over here actually. No way, no issue. That will help you to understand. DNA fragments have negative charge, right? The question I've been asked in 2017. Which gives negative charge to DNA? Which gives negative charge to DNA fragment? We have discussed Iram. Yes.
Yes, sir.
Which gives the negative charge?
Is negative charge because of the phosphate groups, right? That is
Exactly.
given.
Yes, we discussed in starting that the strands are made up of phosphate and sugar, and phosphate gives the negative charge to DNA. Very good. Okay. And what about the histone? What is the charge on histone? Iram? We have discussed that as well.
Histones are positively charged because of
Very
the
good.
amino
Good, good.
acid.
Very good, very good. Excellent. So, one more thing. If there are 999 bases in RNA, that codes for a protein with 33 amino acid, and the base position at 901 is deleted, right? Such that the length of RNA become 998 bases, right? How many codons will be altered? Tell me. How many codons will be altered? Iram, look,
Sir, I didn't understand the question.
look, there is total 999 codons, sorry, 999 bases, right?
Mm.
After 900, 901 is deleted. Then how many bases are there total? How many bases left?
998.
Come on, not 998.
998, right?
999 bases are there. After that, from 901, right? The change take place here. Now it will, the frame shift mutation will be in all these 99, right? So 98 genes, they will be get the frame shift mutation, right?
Oh, okay.
Right?
Yes, sir.
This 98 will be affected after deletion. Look, this is a gene. Start from 0. This is 900, right? Now deletion is taking place here. Deletion taken place here. So how many? Will they be get affected? They won't get affected. This is deleted. Then the rest of 98 will shift to this word, toward this way. Got it or not? You have removed a nitrogen base from here. If you remove the nitrogen base pair from here, then what will happen? These 99 will get affected, but this 900 will not be get affected. Will they get affected? These 900?
Yes.
They won't. Only 98 will get affected, right? So 98 upon 3, because every triplet or every codon is made up of 3 nitrogen base? So what is the answer?
30.
All 33. What is the answer? 33 is the answer. Did you got this thing or not?
Understood.
Very good. Abhinav and Alia, please go through with the previous this one. Okay, even Alia have told the correct answers. Positive on histone, phosphates, right? Very good. So Alia and Abhinav, please go through with the previous videos, right? And cover this concept. We have discussed in this concept. This was actually frame shift mutation, deletion. So next question is, which of the following RNA act as structural RNA as well as ribozyme in bacteria? Right, 23 sRNA. The rest are not structural RNA. These RNA are not found in ribosome. These. Yes, Siram? Are they
Yes,
found? No.
No, sir.
Yes, these RNA are not found in ribosome, and we have discussed that thing. Look, the question: the equivalent of a structural gene, cistron. Cistron is equivalent to a structural gene. Euler conducted the semi-conservative model. I was discussing Iram in the last class. On what experimental organism? Escherichia coli. Which of the following RNA is the most abundant in animal cell? Ribosomal RNA, 80%. The association of histone H1 with the nucleosome indicate DNA condensed into the chromatin fiber, means condensation part is taking place. Condensation of DNA is taking place. The final proof for DNA as a genetic material comes from the experiment of Avery, MacLeod and McCarthy or Hershey and Chase. Which one?
Hershey.
Exactly, Hershey and Chase. In 1944, it became evident that DNA is genetic material, right? Okay. And spliceosome are not found in cells of bacteria. Commonly used vector for the human genome sequence. One thing which I want to tell you, the human genome sequence was done by the BAC, that is the bacterial artificial chromosome and yeast artificial chromosome. Got it?
Yes, sir.
Okay. So is that clear?
Yes, sir.
Okay. So the next thing which we will cover, that is the biotechnology part. That is a fresh topic, so we'll start with that. And Heather, I think you was there in last class.
Yes, sir.
Okay, so today you are late, yeah? It's too late.
Yes, sir.
Actually, take the video part. We have covered 3 things, and it was very important, very important. Lac operon model, we did that, right? Human genome project, we did that, and what else? Lac operon, human genome project,
DNA fingerprinting.
and we did that DNA fingerprinting, right? All are important. Go through with it, right?
Okay, sir.
So now we are going to start with the fresh topic, that is a fresh chapter, that is biotechnology part, right? Very important. So as we are covering on the basis of weightage part, right? So if you people want, then I can share with you that is what is the weightage, right? If you want, I'll share with you, right? What is the weightage of biology, right? So the maximum weightage is of genetics part. I told you 18 question average, right? Now the next weightage is of this biotechnology, right? So you can get 9 to 10 questions from biotechnology as well, both of the chapters. Right? It's easy, it's interesting. So let's start with the biotechnology. So first chapter is biotechnology tools and technique. Fine. So there are, even this question have been asked in medical examination, right? What is the basic principle of biotechnology? So there are 2 principles of biotechnology. The first principle is genetic engineering, and the second principle is chemical engineering, right? Can anybody tell me what is the genetic engineering, what is the chemical engineering? Actually, the chemical engineering question have been asked. What is the
Sir-
chemical engineering or what is the genetic engineering? Anybody of you?
Sir, chemical engineering is like, how do you say,
Storing in non-sterile conditions
yeah,
for multiplication.
Okay, good,
for
good.
products.
And what's about the genetic engineering?
Alteration of the genetic material and then inserting it into the host organism and maintaining it there.
Very good, very good. You guys are smart one. Fine. So genetic engineering is just alteration in the genes, right? Alteration in the genes means, in short, I can say formation of transgenics. Transgenic which have external gene. Chemical engineering, you have made, with the help of genetic engineering, you have made a transgenic organism. Now you want the product of that organism. The process of production of that chemical, that particular product from that transgenic is called chemical engineering, right? For example, previously what we used to do, diabetes was present over there, and because of diabetes, what we used to do, we used to take the insulin from the some animals, and basically from the horse and some other cattle. So the
Pig,
yes,
the pig, right? We used to take it.
exactly. Very good. So we used to take from that, and it is injected. But most of the people, like, there was some problem. First thing, how many animals you will kill? Because every animal have a 200 gram that pancreas, and the 200 gram pancreas have some ml of insulin. So how many animals you will kill? First thing means there was less production. Second thing, the insulin of animal is made up of some different protein, and sometimes this protein used to cause allergy in the humans when it is injected. Second problem. The third problem was ethical issues. Some people never use this animal product, right? And other ethical issues. So because of ethical issues, some people did not used to take it, right? So that was the reason behind it. Now, nowadays, what we are doing, so scientists have taken out that insulin producing I gene from humans. They have picked the I gene from the human cell. Then that I gene incorporated into a bacteria. They cut down the bacterial gene and they incorporated that I gene in the bacteria. Now you made the transgenic bacteria. This process of production of a transgenic bacteria which have insulin gene was a genetic gene. Now, after getting that bacteria, we produce hundreds or thousands of the bacteria. Now we used a bioreactor. In that bioreactor, we multiplied that thousands of bacteria into million of bacteria. Those million or trillion of bacteria produce a large amount of the insulin. We multiplied the bacteria, we collected the insulin, we filtered the insulin, we filled them in injections and marketed it. This complete process was called we collected that part, that injection. This complete process was chemical engineering. One more thing, that is not a biotechnology principle. So these two were the biotechnology principle. Now what we have filled them in injection. Now after filling them, packing in a large packet and marketing them and taking them to the patient, this part was called downstreaming process. So this is also a question, what is the downstreaming process? So marketing them for the human use was a part of downstreaming process. So this was the principle of biotech. Look, I have represented over here how we make the transgenic. So this is a human gene and this is a bacterial gene, right? This is the bacterial chromosome. We have to do nothing with the bacterial chromosome. Actually, bacteria have some extra chromosomal DNA, right? This is called plasmid. You know that. That extra chromosomal DNA, this extra chromosomal DNA this extra chromosomal DNA is taken out from the bacteria. Now, this is a human gene, right? From human gene, we have taken the growth hormone gene, right? We cut this gene with the help of a scissor, and the name of that scissor enzyme, there is an enzyme called endonucleases, right? With the help of endonucleases, we cut down the gene. If you have cut down this gene with the help of endonucleases, you have to use the same endonuclease to cut down this. You cannot use the different endonuclease. One thing which I want to bring in your notice, you cannot cut down this gene from another endonuclease and this gene from another endonuclease. You can't do like that. If you have cut down this gene with a particular endonuclease, you have to use this same endonuclease to cut down this one. Now the endonuclease, with the help of endonucleases, we have cut down this one. After cutting down this one, we have taken out, like now we have ligated it. So these ends are called sticky end. When you will use the same endonuclease, you will get the sticky end at the both of them. These sticky end will get stuck with each other. Why it is, we'll discuss in this class only, right? Now these sticky end will get stuck. Now this plasmid, the plasmid which have some part of the bacterial gene and some part of the foreign gene, this is called recombinant DNA. This is called recombinant. Now this recombinant DNA is inserted inside another bacteria. Now the bacteria which have external gene, that is a human gene, this is called transgenic, or we can call it GMO, genetically modified organism. Now this bacteria will produce human growth hormone. We will extract human growth hormone commercially from here, right? So this was the genetic engineering, and this was the chemical engineering. Is it okay for you? Did you got that point?
Yes, sir.
Yes, sir.
So please take this screenshot. Biotech is very interesting part of the biology. Now come to the restriction enzyme. What are the restriction enzymes? I have already told you that restriction enzymes are called molecular scissors, right? Because they used to cut down the DNA. A very smart question. It is not there in like in your NCERT. How these restriction enzymes cut down the DNA? Any one of you? This question is out of NCERT. I do agree.
They recognize the base sequence and then make cuts at the parts. Is that the answer?
Sorry.
Uh,
Yeah. Please
they
go
have like this DNA sequence, they recognize it and then they make cuts at the specific sequences.
Exactly, that's true. But how does they cut down? What they break actually? Let's make it easy. What they break in DNA when they cut down? They break DNA backbone. Now please tell me.
Sugar phosphate bond.
Exactly. Restriction endonucleases, if you want, please you can note it down. Restriction endonuclease, please note down. Restriction endonucleases, they breaks down the phosphodiester bond. They breaks down the phosphodiester bond. And that's how they cut DNA backbone, and that's how they cut DNA backbone, right? So actually restriction enzyme cut down the phosphodiester bond, and that's how they break the DNA. And yes, you told very very right that they recognize a particular sequence, and that is called recognition sequence. After recognizing recognition sequence, they break down the DNA. Okay. Basically, nucleases are those enzymes which break down the nucleic acid, means DNA or RNA. So nucleases are, we call those enzymes nucleases which break down basically DNA, right? So nucleases are the enzyme which break down nucleic acid, which breaks down nucleic acid, right? Those are called nucleases. Nucleases are of two types: endonucleases means inside, exonucleases means outside. So endonucleases make a cut between at the mid part of the DNA, right? Between 5 or 3 prime, right? They make the cut at the mid part, right? Exonucleases cut the DNA from outer side, right? So endonucleases make an internal cut and exonucleases make external cut, right? So that is called restriction. Look, now what is the restriction endonucleases? Endonucleases make internal cut. The restriction endonucleases make a cut at a specific site, exactly where you want. Endonucleases can make a cut at the any part between this 5 prime end to 3 prime end. At any point, they can make a cut here, 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 or here, right? But restriction endonucleases are those enzymes they make the cut specific exactly where exactly you want, right? So these are called, that is the difference between the restriction endonucleases and normal endonucleases, right? Restriction endonucleases, they are an enzyme that cleave a molecule of DNA at any site where a specific short sequence of nucleotide occurs, right? There are two types of the endonucleases. They cleave the DNA molecule. Exonucleases, they cleave the DNA molecule from outside, the same thing which we have discussed, right? So that was about the restriction endonucleases. Now, what is the palindromic sequence? What are the palindromes?
The words which spell the same both ways.
Exactly. Malayalam, either you study from this end, you read out from this end, or you read out from this end, right? It's Malayalam, and the meaning is also same. The same thing is that, right? Look, when we make the cut with the restriction endonucleases, so they make the palindromic sequence, right? Look, here is A T T. The sequence is here, right? Look, the sequence is A T T. Now in the lower end there is the same sequence A T T, right? So they make the palindromic sequence. The part of the, in the terms of DNA, the part of the DNA which have the repeat, like the same sequence or same sequence and a same sequence at the both of the strand, and that is suspended outside as a single strand, is called palindrome. So this is called DNA palindrome, right? Look, what is the benefit of this one? The benefit of this one, suppose that this is, and why we use the same restriction enzyme? Why we use the same restriction endonuclease? To cut down the DNA, this DNA, and to cut down the vector means plasmid. Why we use the same thing? The reason behind it, when we make the cut on this DNA, what happens? You can see. We have made a cut on this DNA. Here, what is the sequence? The same thing. AATT. Now we have made the cut. It will make the same palindrome here as well, right? Because of the same palindrome, when we will rejoin them, they will make a sticky end, right? So they will help in the rejoining because it will make the same palindrome on this one and the same palindrome on this one. Because of the same palindrome, these both of the DNA part will rejoin easily. That's why we use the same restriction endonuclease enzyme, right? That is the same thing, right? Is that clear? Palindrome is clear? Tell me, guys.
Yes, sir.
Yes, sir.
It's easy. It was easy actually. Now, what is recombinant DNA and vector? Right. Look. What are the vector? Vectors are those which are used to take your desired DNA from one place to another place. Those are called vector. So basically, these are the plasmids. We take out the plasmid from bacteria and we add our desired DNA with this plasmid. Now, this plasmid will take the DNA wherever you want. If you want to incorporate in a plant, they will take to a plant cell. In an animal, they will take to an animal cell. In bacterial cell, they will take to bacterial cell. Wherever you want to insert your desired DNA, these vector will take your DNA to that desired destination, right? So they are called plasmid or cloning vector. Look, this is a desired DNA, the foreign DNA or interest gene, right? We have cut down with the help of restriction endonucleases. Now, this is the cloning vector, right? So cloning vector, again, this is the cloning side. We have cut down this part of the DNA. We have removed this part, right? After cutting down this part, we have removed this part. Now, both we have cut down with the same restriction enzyme. So both will have same kind of the palindrome. Both will join. The joining of takes place easily itself, and then after the joining, we use our enzyme ligase. Ligase is called molecular gum, right? Ligase is called molecular gum. So with the help of molecular gum, we add both of them, right? So now the DNA which is formed, this is called recombinant DNA actually, right? So this is called recombinant DNA. That's how the recombinant DNA formation takes place. Just oh. Suppose that there is an enzyme, restriction enzyme. That's what I was searching. Suppose that there is a restriction enzyme. Don't write on this restriction enzyme. Suppose that there is a restriction enzyme, E. coli, RY13. So this is the name of a restriction enzyme, right? The first term, this is the name of restriction enzyme. Name of restriction endonucleases. What does this E represent? Escherichia, that is the genus. So in the name of restriction enzyme, this is important. This is the question actually. That's why I want to tell you. This E represents the genus of the source, right? This coli, this represents species. What does RY represent? Strain. What is 13? This is the number, means this is the 13th enzyme discovered, right? It represents the sequence or number of enzymes right? Please note it down, this one.
So why is the Hind2 enzyme named as 2 when it was the first enzyme that was discovered?
I'll tell you. Just note down. I'll tell you. Actually, the first and second tell you the sequence. That's how they have been discovered. Look, like you must have seen a very peculiar example. First, you use that one in photosynthesis. You must have seen photosystem second come first, then photosystem first come later. That you must have seen. Actually, the photosystem first have been discovered on the later on, right? And the photosystem actually in the photosystem second that comes when we go through that cycle. The photosystem second come first during the reaction of the cycle, but it have been discovered in the later time, right? So that's how it happened. So whenever we write down the enzyme, we write down the enzyme the way it performs its mechanism, the way it is used, right? Got it? The way it is used. Suppose that there is a 10 reaction and that enzyme come first at the first position. So we will put it in the first position. But there are fair enough chances that should be that must have discovered in the 9th position. So we will write it at the first position, and but its name will be that the 9th enzyme. That's what happened. Note it down.
Yeah, yes, sir.
Okay. Now I will tell you the cloning vector. Look, this is the gel electrophoresis process, right? It is used for the DNA fingerprinting as well. From here, the many questions have been asked. I'll tell you what are the questions. Actually, I have underlined that thing, but the questions have been asked. So, first thing, why we use the agarose gel? Why we use the agarose gel? Tell me.
It can stick DNA fragments to it.
Yes,
And
but
there's
why?
also
How does it perform its function? Means how does it separate the DNA? You are close to the answer. Tell me, please.
so when an electric field is passed through agarose gel, the large fragments, they are not able to transfer from negative to positive terminal as the electric fields move from negative to positive, and the smaller fragments which are light in weight, they move further away from the negative terminal. So that's
Okay.
how we get DNA
Okay.
fragments.
Fine, fine. Look, the purpose of gel electrophoresis or agarose gel electrophoresis is just separate the different band or different fragment of the DNA of different length, right? Actually, the agarose gel is a type of polymer. It's a sugar polymer, carbohydrate polymer. It has small pores, right? A very small pore. So whenever the DNA pass through these pores are that much, the pores are of that much diameter so that the DNA could pass through them, right? But these pores create a kind of the barrier or a kind of the hindrance when DNA start moving from one end to another. They provide less hindrance for the smaller one and the more hindrance for the larger part of the DNA, right? That's why the smaller part of the DNA, they get the less hindrance or less obstacle, so they travel faster, and the larger part gets more friction or more uh, hindrance, then their speed slows down, right? So in DNA fingerprinting, what exactly we do? We use a gel electrophoresis process, agarose gel electrophoresis process. In this, we have a tray kind of the structure, this tray kind of the structure, right? In this tray kind of the structure, there is a gel that is called agarose gel. Agarose gel commonly used as a matrix. Agarose gel, it is a natural polymer extracted from seaweed. Seaweed means brown algae. So basically, we extract it from the brown algae or seaweed, right? It is found in the walls of seaweed, right? Cell wall of the seaweed. Now, uh, we have put the part of the DNA. So we have got a DNA. We have broken down the DNA with the help of restriction enzyme. Different fragment of the DNA of different fragment length have been broken down. Then DNA have the negative charge. We put a positive charge over here. We pass a positive charge over here at this point. When we pass a positive charge over here, then after passing the positive charge, this DNA will start moving from negative to positive towards in this direction, right? When DNA starts moving, the larger fragment remains at the behind, and the smaller fragment they get arranged at the forward side because of the hindrance I have told you, right? Then what happened? Different bands are created. After the creating different band or arrangement of the different band with a different length, we separated fragment. We separate the fragments. These fragments are stained with the dye ethidium bromide, right? Ethidium bromide. The name of dye have been asked many times, ethidium bromide. And after the staining DNA with the ethidium bromide, we put a cellulose, nitrocellulose film on this agarose gel. What film? Nitrocellulose film or nylon films. We put a film over this agarose gel, and these part of the DNA or these DNA get stuck with that nitrocellulose film. Then we take out that DNA. We separate DNA from this gel. The process of separation of DNA fragment is called elution. This question have been asked many times. Ethidium bromide have been asked, elution have been asked, right? After the elution means we have taken out the DNA in that nitrocellulose film or that nylon film. We drop ultraviolet rays on the this DNA. When ultraviolet rays fall on the DNA, this DNA band, we get a particular kind of the light. What kind of the light come out? That is also a question have been asked. It's there in NCERT. Very simple. When you stain the DNA with ethidium bromide, after staining the ethidium bromide, you put a light, ultraviolet light on this DNA. What kind of the color will come out? It is there in NCERT. This question have been asked.
Sir, orange colored band.
Very good, excellent. Orange color will come out. That is also a question. So sometime question directly come from NCERT. 70% question they directly come from the NCERT. 70-80% sometimes, right? Sometimes it may be 30% from outside, sometimes 20%. But mostly question they come from the NCERT. They may be directly or indirectly from the NCERT. Now the DNA fragment is taken out and joined with the vector. That's how the ligation takes place. So I please take the screenshot of this one. Clear?
Yes, sir.
Now come to the cloning vector. Very important. Okay. So what are the characteristic of vector and what should be the in a vector? Look. Now first thing. Actually now your question. Hint second. Actually the hint second was the first restriction enzyme to be discovered, but its functionality was discovered 5 years later. Means the sequence on which it is working that have been discovered on the later year. So it was discovered first, but we put the name hint second, right? On the basis of because its functionality was not discovered when the enzyme was discovered. Means the sequence on which it is working, the sequence have been discovered 5 years later. So then we given the name hint second to this one, the first discovered enzyme, and hint first who were discovered after that and its functionality was known. We given the name hint first to that one. Got it? Got my point?
Yes, sir.
Yeah. So actually the hint second was the first enzyme, and that you need to remember. Sometimes they ask which is the first restriction enzyme discovered. So the first restriction enzyme discovered was hint 2, but its functionality was discovered functional sequence. The sequence on which it worked means functionality was discovered 5 years later. Okay. So what is the vector? We have discussed what is the vector. What a vector should have means what the qualities a vector must have. So these are the feature of a cloning vector. So before going that deep into, I want to explain this one. This is a cloning vector. The name is PBR322. It has been used like for the number of the purposes in human being and for the experimental purpose, right? So PBR 322 is given your NCERT, and in PBR 322 there is a site that is called this one. This is called origin of replication, or IC, right? Origin of replication. Origin of replication is that site from where it is written over here. Origin of replication. Origin of replication is that point from where the replication of a plasmid starts. Plasmid is semi-autonomous for its replication. Means it can replicate itself. Why semi-autonomous? Why not complete autonomous? The DNA is autonomous. DNA can replicate itself. Its replication of DNA does not depend on anything else. Replication of DNA depends on DNA itself only. So that's why we call replication of DNA is autonomous, either of its bacterial DNA or of its eukaryotic DNA. But the replication of the plasmid is semi-autonomous. Replication of mitochondrial DNA is semi-autonomous. Replication of chloroplast DNA is semi-autonomous. So 3 DNA, external DNA, CLPT, chloroplast DNA, mtDNA, mitochondrial DNA, and plasmid. They are semi-autonomous replication basis. Why? Why not autonomous? Tell me, guys. It's a very smart question. Abhinav, Alia, Heather. Iram. Look, very simple. It have all capability of replication itself. But for the replication, it requires certain enzyme. Those enzymes are coded by DNA only, nuclear DNA. So nuclear DNA code enzyme for the replication of this. It require the enzyme, and those enzyme coded by, so it is dependent on nuclear DNA for enzyme. That's why it is not fully autonomous, right? If you want, please note it down. Note down. Plasmid, comma, mitochondrial DNA, in bracket mtDNA, comma, and chloroplast DNA, CLPT DNA, and chloroplast DNA, CLPT DNA. Is semi-autonomous for replication. Is semi-autonomous for replication because the enzyme which takes part in their replication, because the enzyme which takes part in their replication are coded by nuclear DNA. Hence they are partially dependent on nuclear DNA. Hence partially they are dependent on nuclear DNA for replication. Okay. So origin of replication is that part, or IC, from where the replication of plasmid starts. Origin of replication have one more property, one more role. What is the second role of origin of replication, or IC? Now you have to tell me. Replication start from here. Guys, come on, tell me. Or IC also define the number of copies, it will make. Will it make? After the replication, it will make 100 copy, 50 copy, 20 copy, 30 copy. It will also be, number of the copies will also be decided by your IC. Your IC have two functions. First, the replication will start from here. Second, it will decide the number of the copy are being formed. First thing. Second, now this is hind, third, echo R1. These are the site where these are called restriction site. These are the site where restriction enzyme will work. Third, BAM H1. BAM H1, this is the site. What is that? What we call that? BAM H1. Come on. No? BAM H1 is an insertion site where the new part of the DNA will be inserted. This is called cloning site, right? So BAM H1, it is called cloning site. Why? Let me write down. Cloning site. Here, the foreign DNA will be inserted. So DNA is inserted, means this part will be removed from here. Please try to understand. This BAM H1 is the part which is called recognition site, right? Cloning site. Here, this part will be removed and new DNA will be inserted here. Please try to understand. In this BAM1 part, there is a gene, beta galactosidase producing gene. Beta galactosidase is a type of chemical that is produced by this gene, the gene present at the BAM H1 site, right? When you will remove this site, this part of the DNA, you will insert a new DNA, then beta galactosidase gene will be removed from here, and the functionality of beta galactoside gene will stop. This process, so due to insertion of a new gene, the functionality of previous gene stops. This is called insertional deactivation. Is that clear or not? Please tell me, guys. It's very important.
Yes, sir.
Insertional deactivation. Got it? Okay. Again, you can see tetracycline resistant gene, MP cycline resistant gene. Actually, these are the genes. These are called marker genes. These are the genes which protect bacteria from the tetracycline antibiotic or MP cycline, which is also antibiotic. So TET-R or MPR, they are the marker gene, and they are used for the their function is to protect bacteria from the antibiotic medicine. Why we call them marker gene? Got? Try to understand. Suppose that what you did, along with the foreign gene, you have incorporated tetracycline gene, right? Now, the tetracycline gene is present in the bacteria, which is recombinant, right? But non-recombinant do not have tetracycline gene. So you have to differentiate from your bacteria, means recombinant bacteria, from the non-recombinant one. So what you will do? You will put the tetracycline in that medium. What will happen? Non-recombinant will die and recombinant bacteria will not die, right? So they will work as a marker. They will mark your recombinant bacteria. That's why these genes are called marker genes. Clear? Look, I have written over selectable marker.
Again.
Yes.
Can you explain this one again? Tetracycline and
Yeah, sure. Look, suppose that, for example, what I did, I have a plasmid, right? Simple plasmid, and I have inserted an insulin gene that plasmid. Along with the insulin gene, I have also inserted a tetracycline gene, right? So now this plasmid have been incorporated in another bacteria. Now wherever my gene will go, wherever my plasmid will go, means suppose that I have thousands of bacteria. I have incorporated thousands of the plasmid, but all bacteria will not take my plasmid. So for sake of example, 600 bacteria out of those 1000 bacteria, 600 bacteria have taken my plasmid, right? Means those 600 bacteria which have taken my plasmid, they have also taken indirectly my tetracycline genes. Now, out of these 1000 bacteria, I want to know which are the 600 bacteria which are the GMO or which are transformant. So what I will do, I will put the tetracycline chemical antibiotic. Because of tetracycline gene, resistant genes is not present in non-recombinant. Those non-recombinant will die and my bacteria, which are the recombinant bacteria, they will survive. That's how I have marked my recombinant bacteria from non-recombinant, right? That's why we call them selectable marker, because they have marked my bacteria. They have selected my bacteria. They have selected recombinant bacteria. Is that clear?
Yes, sir. So the antibiotics tetracycline and the ampicillin will be inserted by us only.
Normally, there are two things here. Look, suppose that we have selected a plasmid which already have these genes. So we need not to select. Marker gene is already present. We need not to do that, right? So normally the genes, we select the plasmid which have tetracycline or ampicillin, any of the resistant gene, we select that, right? And we incorporate them. So the bacteria which get these plasmid, so they will not die in the presence of ampicillin or tetracycline, but non-recombinant will die. That's what happened.
Okay, sir.
But nowadays, one more thing. Now what we are using? Nowadays, we are not using the tetracycline gene. What we are using, so in insertional site, there is a beta galactosidase, right? Now what happened? This beta galactosidase, when we insert our gene, beta galactosidase gene is removed, right? So the bacteria which are the recombinant, they will not produce beta galactosidase, right? Now, when we will put the when we will drop the this ultraviolet rays, because those bacteria which have the beta galactosidase gene, means non-recombinant, they will produce fluorescent color. But those which are the recombinant bacteria, they will not produce fluorescent color. Why? Because the recombinant bacteria do not have beta galactosidase gene. They will not produce beta galactosidase. Absence of beta galactosidase, they will not illuminate. They will not produce illuminance. Got it?
Yes, sir.
Or sometime we use fluorescent gene. Sometime we use fluorescent other genes which produce fluorescent color. So nowadays we are using the fluorescent gene for the marker in place of these antibiotic resistant gene. Okay. So selectable marker, normally they are the antibiotic resistant gene. To link with the alien DNA, cloning site, to link with the alien DNA, it have recognition site commonly used by the restriction enzyme. Chromogenic substances also added here. Where? In recognition site, here, this one. In its insertional site, cloning site, we also use the chromogenic substances, right? We use the chromogenic genes, which produces the chromogenic substances, right? As a marker. Insertional inactivation of beta galactosidase takes place, hence cloning site, hence cloning bacteria do not produces the blue color in ultraviolet rays. Okay, one more thing. The vectors which are used for the plant as well as for the or for the animal. Normally we use AGBT vector. This question have been asked. Normally we use the AGBT vector for the dicot plant. Now what is the AGBT vector? Can anybody tell me what is the AGBT vector or what is the AGBT bacteria? It's important. This is also important. What is the AGBT bacteria or what AGBT vector? Look, there is a bacteria. No name is Agrobacterium tumefacium. What is the function of this bacteria? This bacteria causes root tumor in dicot plant. Okay. These bacteria causes root tumor in dicot plant, means root cancer in dicot plant, right? Inside the AGBT, inside the Agrobacterium, there is a plasmid. We call that plasmid AGBT. Please try to understand. There is a bacteria, Agrobacterium tumefacium, that causes cancer in the roots of dicot plant. In that bacteria, there is a plasmid. We have named that plasmid AGBT.
So
AGBT plasmid. Sorry.
Isn't the name of the plasmid TI plasmid, something like that?
Look. Try to understand. That AGBT. So we have named that AGBT, right? That plasmid. Now, after that, actually, that plasmid have In short, AGBT is that plasmid name, that plasmid AGBT vector, right? AGBT vector means Agrobacterium tumefaciens plasmid. That plasmid have a DNA, that a special DNA segment that is called tDNA. Actually, this tDNA, which is present, which is a part of that plasmid, causes tumor. When we use this bacteria, when we use this plasmid, we cut down this tDNA. Means we remove the cancer causing part of that plasmid. Now we call that plasmid TI plasmid. Clear? We call that plasmid TI plasmid.
Yes.
Now this TI plasmid is used as a vector for plant to transfer gene in plant. Is it clear or not?
Yes.
Yes.
Okay. Retroviruses, they causes cancer in animal cells. Retroviruses means they are the viruses where the gene is RNA, right? The viruses which are made up of RNA plus protein, we call them retrovirus. For example, HIV. The question I've been asked in medical. HIV is a retrovirus. Any other example of retrovirus?
Tobacco mosaic virus.
Very good. TMV. TMV is also a retrovirus, right? So the virus which are made up of protein plus RNA, we call them retrovirus. These retrovirus have cancer-causing gene. We remove cancer-causing part of that virus and we use them as an animal vector, right? That's how the vectors are being used. Tell me, is it clear or not?
Yes, sir.
Clear? So this was the plasmid, and what else is left? The rest we'll discuss. Actually, the three topic have left over here. Process of recombinant DNA formation. That is very, very, very important. PCR is also very important. Number of time have been asked. Bioreactor. It's okay. Right. So next class we'll discuss these rest of these three topic and we'll complete this biotechnology part. And now, if you have any question from this chapter, please ask. Please ask me if you have any question in this biotechnology part, biotech part.
So the other pink part in this cloning vector is also the insertion site.
No, actually, this part is tetracycline. Actually, the picture was not clear, so I have made a line. This part is actually this BAM part, and this lower part, this is tetracycline resistant part, and this is empty cycline part. PBR have been asked as a question in medical examination multiple times, many times, and you have to remember the site as well. Diagram based question also come from the PBR. You will see the question. And what I say, one more thing I forgot to tell you. You must have got the previous year question paper PDF form of Arihan,
Yes.
and please start doing question. At least you have very limited time. If you can do more, then it's okay. But minimum, what I expect from you, you have to solve 2018 onward questions. So the questions which are from the 2018 onwards. So you have to do all those questions.
Okay, sir.
And the best thing is that with that PDF, explanation is given with that PDF. Otherwise, in other publication, the explanation is not there. Normally, people go for this DNA, this one, fingertip and all that, but explanation is not there. So go through with and solve all the questions. And in case if you are getting any problem, any doubt in the question, write it down there, right? Ask me in the next class. You're most welcome with your questions, right? Any doubt?
No, sir.
So first, I want the response from the new one. Abhinav, got everything?
Yes, sir.
Is that pace is okay?
Yes, sir. It's fine.
Okay, fine. Alia, what about you? Are you okay with my pace? Are you getting everything? Okay. Very good, very good. Great, great, great. Heather is already there, like his old student. Iram is already there. Yes.
So our previous year questions enough for practice when it comes to biology?
Look, what you have to do, you have to always take this screenshot. Whatever I am teaching you, previous year question, and NCERT. Believe on me, you will be able to do more than 85% question. NCERT, don't run here and there. Believe on me. NCERT, whatever I am teaching with you and previous year, that is sufficient for you. Don't go. And in case if you can take more pain, go for the fingertip.
Okay.
Fingertip. If you have fingertip, you can go with it. But that should be the fourth option. That should not be third option. That should be the fourth option. You have to take care. Actually, the time management is the best thing, right? You have very like a limited time. On that limited time, you have to take care of physics, chemistry, zoology, botany, all things, right? So you have to give equal importance to all of that, right? So for that, what you can do it with the full of your energy, right?
Okay, sir.
So see you guys in next class, right? Bye-bye. Take care. Have a
Bye,
good
sir.
day.
Thank you, sir.
Welcome, welcome.
Excuse me, sir. Sir, what
Yes.
is the function of ROP in this?
This one.
ROP. The yellow colored part.
Okay, this one. PUV genes. Actually, this is also a site for the this one, insertional site.
For the new DNA.
Yes, but actually the insertional site can be more than one. Even one more important thing, that that can you get the question? Even the origin of replication can be more than one, but there will be only one major ORIC site and one major insertional site.
Okay, sir. Thank you, sir.
Any more question? No. Okay. So see you in next class. Come with more question if you have, right? So and go through with previous year question. Okay.
Okay,
Bye bye.
sir.
Take care.
Thank you, sir.